Mitosis and meiosis review video. I would encourage you before you get started to draw out 5 beautiful, perfect circles. And then once you have 5 circles done, then get back to the video. So go ahead and pause it, draw those circles, and get back to the video. All right, this unit is focusing on 2 different types of cell division. This is looking at complete division, known as mitosis, and then division where we're going to have half the DNA. A simplistic view of looking at this, mitosis really refers to what happens in our body almost every single millisecond. It's dividing cells. It's making cells for the ones that are damaged or the ones that are used up. Because our human body naturally kills off cells on purpose to prevent cancer. Because if a cell lives too long, there's a greater chance it could be messing up. So what we've done is we went from 1 cell to 2 cell. This is known as cloning. Okay? Now, ideally, this is known as daughter cells. So when I say daughter cells, this original cell right here has all the chromosomes. It's known as diploid. Diploid refers to di, which is 2, so this is a 2N cell. Now, once a cell makes a copy of itself, it will then make another 2N cell as well, another diploid cell. For a brief time period, this cell is going to have double the DNA. Because it's simple division, you take double, you divide by 2, then you get back to the single amount. Now, meiosis is really referring to the only spot in our body where this happens is the male's sperm and then the female egg. Okay? This term is known as haploid. Haploid cells have half the DNA. Okay? Now, if we're talking about numbers, it's really easy to get these confused. This guy over here has either 23 pair or a total of 46. I mean, it's the same thing, 23 pair or 46. This sperm over here has just 23. Okay? Not pair, but 23 because it's known as haploid. Haploid is distinguished as just N. Okay? Now, when you take this guy plus this guy and you bring them together, this is where we get people. Okay? We are diploid, meaning we have all of our chromosomes if we're lucky, and that means we have 23 plus the 23 over here gives us 46. That is us, okay? Now, this unit is about how this cell goes from 1 to 2, and then we have meiosis, which goes from 1 to 2 to eventually it will then go to 4. And then the woman's system's a little bit more complex. There's some similarities and differences that we'll be going over. So I said you need to start off with 5 circles. This first circle is going to cover what the cell does 90% of the time. So 90% of the time it does interphase. Now, to help you out on the test, what I would do is I would write this out at the very beginning. IP, I-P-M-A-T, IP-mat. Okay? Interphase, prophase, metaphase, anaphase, telophase. Okay? Now, it's very important for you to recognize and distinguish this is just normal cell activity 90% of the time. All right? P-M-A-T is actually mitosis. Okay, actually mitosis. This does not include mitosis. This whole thing from start to finish is called the cell cycle. So the cell goes from interphase, prophase, metaphase, anaphase, telophase. Now, if I were to look at a bunch of cells, I would be able to distinguish 90% of them are probably in interphase. Okay? Now, interphase towards the very end, when it goes, I need to divide, it's going to double up its cells. If you imagine the nucleus inside, okay, here's our nucleus. We drew it nice and big. If this is going to be 1 chromatin, okay, and then here's another chromatin as well, and then let's get another one in there, and then here is another chromatin, okay? So here's our chromatins. We need to double them up, okay? So to double them up, we just make copies, all right? Identical copies. That way we have twice as much information. Now, during this phase, you might see in books where it says G1, That refers to growth phase, and then the S refers to synthase, which means to create the DNA, which that's what happened here. And then we have G2, which means grow again. So this is interphase. Our next phase is going to be where we actually start mitosis. This is going to start off with prophase. Now, as far as Latin go, pro refers to, if we use our Latin skills, pro refers to as first. Okay? Which that kind of makes sense. Pro first. So therefore, this is our first phase. During this phase, we have our nuclear membrane dissolve away, break down. Okay? Now, as this nuclear membrane is dissolving away, our chromosomes, actually chromatids, are kind of condensing. They're actually becoming chromosomes. Now, over here, they're kind of spread out. They're just like taking their time because they're being read off constantly. When we divide them, they need to get a lot smaller and what's known as condense, okay, or supercoil. Okay, so here they are. Now, we also have these other things, these little alien-like structures. These little alien-like structures will go to opposite poles, and these are where we'll have our spindle fibers come out. And these spindle fibers are like aliens. They're going to rip apart our DNA. Now, the next phase up, so we have prophase. If you look through our system all the way through, okay, we got 5 circles, 1, 2, 3, 4, 5. We are right in the middle. Okay, now this transcribes really nicely to metaphase. Okay, metaphase is probably the easiest one to see. Metaphase refers to, in Latin, if we were to use our skills again, meta is the middle. Okay, and then what happens here is our chromosomes line up on the middle. That's a poor way of saying it. A better way of saying it is the equator. So imagine these guys breaking free and just lining up in the equator. Okay, now these little guys off to the side, they've done quite a bit. Okay, they've gone to opposite poles, and those alien-like structures that I was referring you to are starting to come out. So I want you to imagine these alien-like structures making their way out, and they're going to cause some havoc. Good havoc, but they're going to cause some havoc. Okay, so here they are coming out. Where they're going to attach themselves on these guys is going to be in the center. Okay, so a way of saying it is going to be the centromere. So centromere right here. Now, since we're right here, it's an easy way to transition is going to be looking at our 3 parts. Okay, what we have is, if you look at a single one of these, we have one side going this way. Okay, and then we have the identical side. Okay, known as sister chromatid, coming out this way. The 2 sides are identical. So if I were to draw a line right down the middle, we'd be able to divide them up nicely. Okay. Okay. This side over here is identical to this side over here. So if we have a gene right here, that means that gene is right here because we got 2 of each. They're held together in the center by the centromere. Okay, so it's holding that 2 sides together. This is where the spindle fibers are going to attach themselves to rip it apart, and then pull the chromosomes to opposite poles is the proper term. Now, after metaphase, if you look at our IPMAT, I-P-M-A-T, we did interphase, prophase, pro means first, metaphase means middle, and then we get to our next phase, which is anaphase. Okay. This is one of those phases that it's really hard to figure out where the anaphase actually starts because it's a transition. You can't think of this as the cell goes, stops, and goes, all right, ready for the next phase, and does this phase over here. It kind of is a flowing movement. And what this would look like would be our chromosomes pulled to opposite poles. So that guy got pulled over there. This one was in the equator, now it's pulled over here. And then this one over here is pulled over here. Okay, so opposite poles. Now what's pulling them apart again? Those spindle fibers. Okay, so here are those spindle fibers at. And we still have to draw those guys coming out. Okay, now this is the anaphase. Now, when we transition to the next phase, telophase, let's go ahead, write down our Latin meanings. So metaphase, anaphase, and then we're off to telophase. telophase, Latin skills. For this one again, telo means in Latin final. Okay, so this is our final phase. This diagram is actually not drawn properly because I have a single cell. Telophase starts off with a single cell, and then it's going to go to another phase called cytokinesis. So let's first just draw what it looks like in here. Okay, what it looks like on the telophase level is we have our chromosomes on opposite corners. Okay, now while they're over here, though, something else very special is happening is they're starting to get their protection. Okay, their nucleus is developing on each one. So this side will then also have its independent nucleus as well over here because we want two cells. Okay, so this is what it looks like as it's pulling. Now, apart from us developing two nucleuses, we're going to like rip apart this cell in half too. Now, what that kind of looks like is it looks like I'm going to draw it kind of in the middle, the division. Okay, I want you to imagine the cell just being ripped in half. Okay, and then on this side over here, this is where we'll have our nucleus over here, nucleus over here, and then inside we'll still have our DNA as we start to separate it completely apart. There we go. Yeah, nice. Now, some other terms that we have here is this is referring to cytokinesis. All right, cytokinesis. C-Y-T-O-K-I-N-E-S-I-S. Cytokinesis. Now, what is this? The division of the fluid inside. Okay, called cytoplasma. Now, you can't just think that it's just fluid and the DNA. We also have all these other wonderful organelles inside that we talked about and studied. Right next to the nucleus, you might be able to remember, is where we have our endoplasmic reticulum. And then we have the rough ER up here, and then we have smooth ER over here. Then we have the Golgi apparatus over here. We're going to have some mitochondria over here, the powerhouse of the cell over here. Those things are also being divided because remember, we want a final cell. And then so over here we can go on mitochondria and just all of those organelles that we covered, that would be also divided up nice and even. If it's not divided up even, the cell is just going to die. And as it's dying, the body's going to come around and get rid of itself. Okay, so this is referring to cytokinesis, refers division of the cytoplasma. Okay, now our goal is to go from one cell to two cells. Okay, so looking at the whole picture, oh man, I made a mess. Looking at the whole picture, we go from 2N to actually almost 4N. So we have double our DNA. And then we have double our DNA, and now we're starting to divide it up so we have our original DNA over here. So this guy over here is 2N. This one over here is 2N. That's where we started. And you might remember us drawing that out over here, 2N, 2N. This is the whole phase. Remember, where students mess up is interphase is not a part of mitosis. Mitosis is PMAT. And then we can also put a C right there for cytokinesis, all right, division of the cytoplasma. Now, getting to the other side is going to be our meiosis, okay? So for our meiosis, what I would encourage you to do is draw this. See if you can get those nice pretty circles. All you have to do is get a stencil. And then after you draw it, then go ahead, hit start, and get started again. Okay, so we have two processes over here. The first process is sperm. Oh, what's done? Spermatogenesis, the production of sperm in the testes. Okay, so the testicles will have all of their DNA, 2N. The only part in the male body that will only have half its DNA is going to be these guys over here. So this could be spermatogenesis, but I actually drew, oops, this is actually a better representation of oogenesis. Hopefully you caught that. You might notice these circles right here are a little bit smaller, okay, which that's refers to oogenesis. If you were to see 4 perfect circles, then it would be spermatogenesis. But if you remember, down here, give you a little heads up, we're going to get a term. 3 of these guys, or ladies, are polar bodies. Okay. Now, notice that we have 2 divisions. That's what I really want you to pick up on. This one right here is our first cell division. Okay, so if that's our first cell division, Therefore, this one right here is going to be our second cell division. So we got to do 2 cell divisions, all right? You can draw that out really simple. How you draw that out? Just draw circles, guys. Go 1, 2, right there. That's what we did. The only difference is we added another circle here because there's a little bit more important things happening going on, all right? Now this division, our goal is to get down here to have half the chromosome numbers. So when I say half, I say haploid. Okay? So each one of these will have half the chromosome numbers. Another way of saying it is this one will be N, this one will be N, N, N. We'll start up here with 2N. Okay? So if we go from 2N to N, if you think math-wise, if we double up our material, and we only double up our material, half of it will go here, then half of it will go here, then we can't do another division because we can't quarter it up. So what we have to do is we go from 2N to where we have to double, okay? So I want you to imagine if I were to draw in at the very beginning, we just have 2 of these guys, okay? If we have 2 of these guys, and again, this is a very simple organism, like bacteria is even more complex than this. If we go from 2 right here, that means this one we need 4. 1, 2, 3, 4, okay? Now, what we have over here is we still have metaphase, okay? So if I were to draw this entire thing out, you can imagine we have our spindle fibers coming out. The spindle fibers will make their connection and pull these guys apart. And then after that, we will then start to separate them out, okay? So I want you to imagine they're being pulled to opposite sides, and then they're going through the whole process, and then half of them will go over here, while the other half go this side. And what that looks like is like this, boom, 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 okay? Now, the following phase will be our second cell division, all right? Now, you take 2 here to divide it up nicely. 1 goes over here, then 1 goes over here, okay? So this one right here can go here, and then this one right here goes here. And we do that for the other side as well. Right here, okay? So we start off with one that had 2 chromosomes, and now we end with a single chromosome. That means if I were to take this egg right here, and if I were to take this egg right here and combine it with a sperm that has 1 as well, and we're to put them together, what we're going to have is we're going to have what's called a zygote, known as a fertilized egg, that will have both of them in there. Okay? So therefore, we'll get to something that is known, as 2N. All right? Now, some other terms in there. We got a bunch of terms. So we have spermatogenesis, okay? And I'd encourage you to say these out loud as well. Audio helps out with memory. Spermatogenesis, formation of sperm. Oogenesis, formation of the female egg. Then these guys right here and this one right here have the same name, gametes. Gametes refers to one of the sex cells. Now, if I were to look at a human's DNA, it's much more complex than this because remember, we said the human had 46 chromosomes. When I say 46 chromosomes, I mean 23 pair. And what this is, you can look at each human's DNA for like $200. This is called karyotype. This is the activity where you cut out all those wonderful little circles, or not circles, those DNA, and you got to pair them up with homologous chromosomes. Now, how we always pair it, and this is the way we do it, we go from largest to smallest, okay? So I want you to imagine largest to smallest. Therefore, if I had all this DNA spread out, I'd find its complementary pair, and I'd pair them up. So if we got a tall one right there, that would be our chromosome number 1. And then this little guy right here would be chromosome number 2. And then we get this guy right here, this would be chromosome number 3. And notice that I'm giving them homologous pairs, okay? Now, to draw them out a little bit faster, I'm just going to rock it out pretty fast. So if this is our chromosome number 1, this is our 2, this is our 3, and we have 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, woo! Tres más. 21, 22, and then down here we have our 23rd. Okay, now I said that with an emphasis because this one right here is very special. Okay, this one right here determines if you will be a male or a female. Okay, this one determines that. Now, this one I drew two of them the same size. Okay, if you're looking at that, you should be able to tell me that this person right here is a female. That is referring to their XX chromosome. So the X's are all nice and big and powerful. The Y chromosome is pretty pathetic. Okay, everybody, male and female, we all start off as females, and then after a while, like a week or so, 2 weeks, this Y jumps in and starts doing its job. Okay. Now, if I was looking at the chromosomes, our 23rd is our sex chromosome. That means every single one of these, 1 through 22, are called autosomes. These autosomes are basic characteristics that male and females have. Okay, so like hair color, eye color, skin color, height, all these traits are right here. Now, this karyotype chart, we can also determine chromosome mutations, and we're going to be covering one next 2 units from now. This mutation is very, very challenging. It has a wide spectrum, and that's where on the 21st chromosome there's an extra one known as a trisomy. Now, notice our homologous chromosomes. Homo in Latin means the same. Chromosomes means a combination. Homologous chromosomes. Wonderful terminology, okay? If I were to look at both these together, right here, this would be 2N. If I were just to look at half, right here, that little guy, this one right here would be just N. So this is our diploid. This is our haploid. Okay. Now, other things on the test, there's going to be a wide range. What you should be able to do also is I will give you some math. Things that you'll need to be able to do is look at a cell and determine what phase they're in. So if I were to give you a cell that looks like this, okay, you should be able to tell me what phase the cell is in and what's happening. This one would be anaphase. Okay. Now imagine I give you tons of cells. So what you have to be able to do is average and turn that average into percents. I only care about the final result of the percents. Okay. So here is a crash course. Okay. There's other things on the test, of course, but here is a rough idea of what will be on the test. As always, I will have a review session early Tuesday morning or the day of the test at 7:40 a.m. So it'll get you in nice and early, and then we'll go over some terms. You have the study stack cards online. You have your packet. You also have the YouTube video that I'm presenting right now. So you have tons of material. Now let's pretend that you don't get everything. Other things that you can do, I'm just going to write it on here. GTS. Okay, Google that stuff. If you're really wanting to drive yourself to get in this material, you can type in Google mitosis quiz. Boom! You'll get a quiz on mitosis. Google meiosis quiz. Boom! And a lot of these quizzes, you just select the option at the very bottom of the screen. You select show me the answers, and it'll grade it, and you can go from there. This is about you figuring out what you need, and it's about time. I know all of you guys can do it. I believe in you. Let me know if you have any questions. Thank you.